Okay, so real quick review from yesterday. We talked about the importance of compartmentalization yesterday. Okay, that is that in a eukaryotic cell, there are the organelles that are labeled on your diagram. Each one of those organelles has got its own unique environment inside that has the conditions that are conducive to doing whatever job that particular organelle does. Okay, we talked about how that makes the eukaryotic cell more efficient, which is why they are not only larger, but can become specialized, which is a precursor to becoming multicellular. All right, so now we're going to pick up here and we're going to hit the ground running with what do all the parts do? Okay, so you actually have all the functions. Now, as we go along, okay, I'll kind of underline and write a couple of things on here. Okay, whatever I underline or write on here, that's going to be like your bare minimum for I need to know that for sure as the function or some important detail about that particular structure. Okay, and then from there, we'll go into cellular transport and we should be able to get this whole thing done today. All right, so first off, first thing we want to talk about is the role that DNA plays in the cell. Okay, so first off, I'm going to scribble that part out because I don't like how it's worded. Okay, so DNA is used to make materials. We've talked about that before. DNA is the instructions that tell your cell how to build things, in particular proteins. Okay, so that's the job of DNA. Okay, the nucleus stores the DNA. Okay, so it says here, in the environment of the nucleus, DNA in the form of chromatin is stored. The chromatin part, probably, I wouldn't worry about that part. Okay, I'm going to get rid of the underline there. DNA is stored. Okay, so the purpose or the job of the nucleus is to store DNA. Okay, it is not the brain of the cell. Okay, if you write brain of the cell, I will become enraged. Okay, that is not what the nucleus does. Okay, a brain implies like decision making, active control over something. The nucleus doesn't do that. Okay, all the nucleus does is make sure that the instructions for building things are safely contained in one place. If you want an analogy for the nucleus is a library. Okay, that's a better analogy than brain. Okay, it is a library. All of the information needed to run your cell, make everything it's going to need, is stored there. Okay, now within the nucleus, not only do you have all of that DNA, but you also have this other kind of dense structure in the middle. Okay, that is the nucleolus. Okay, now when we were talking about labeling the cell diagram, we said nucleus and nucleolus are really the only two I'm really worried about spelling because they're so close. Now the nucleolus, if the nucleus is the library, the nucleolus is the librarian. Okay, it knows exactly where all of the information is. It knows every page of every book in the entire library. Okay? Your nucleolus knows where every single part of your DNA is. So if your cell asks for it to make insulin, it knows what book, what page to go and copy. All right? So what the nucleolus does is it actually copies DNA to control protein synthesis. Okay, because your nucleus, while like a library, is not a lending library. Okay, it is not letting people check out the books and take them home, because it knows it'll never get the book back. Okay, so what it does instead is it finds the appropriate part of the book and copies it and sends the copy out. Then it doesn't worry whether it gets the copy back. Okay, it still has the original. Right? So what you need from that slide, DNA is used to make materials. The nucleus stores DNA. The nucleolus copies the DNA and controls protein synthesis. It doesn't actually do the protein synthesis, it just makes sure that information goes out to the ribosomes. They're the ones that do the protein synthesis. Okay, so we're going to get into a little bit here of how the process of protein synthesis works. I'm going to explain it in a little bit more detail than you need to know it, okay? But I'll tell you how much you need to know, okay, in terms of how protein synthesis works. Okay, so the nucleus, or nucleolus, sorry, as we said, produces RNA. That's a copy of the DNA. Okay, and that is the instructions to build things. Now, if you remember back to Science 9, you know that DNA is two-stranded, right? There's two strands of DNA in a DNA molecule. What your nucleolus does is when it needs to copy the DNA, it uses a special enzyme called RNA polymerase. You don't need to know that, okay? But that's the name of the enzyme. And that enzyme pulls the strands of the DNA apart. They'll go back together. It's not permanent damage or anything like that. It just pulls them apart. And it copies one side. Why is one single-stranded copy of only one side of your DNA good enough? Why doesn't it have to copy both? No? Okay, last year in Science 9 you learned that in DNA there's those little base pairs, okay? A always goes with T, so adenosine always goes with thymine, okay? And guanine always goes with cytosine. C always goes with G, okay? They're complementary, the sides are complementary, okay? So you know if you have an A on this side, you have a T on that side. If you have a C on this side, you have a G on the other side. So you only need to copy one side. Okay, because the other side will be the reflection of it, so you don't need it. So in order to make a protein, one side of the DNA gets copied, and that makes single-stranded genetic material called RNA. The RNA can then leave the nucleus. Okay, it'll go out through the nuclear pores. And it will merge with a ribosome. Okay? Ribosomes are what make the protein. Okay? So they are like a construction site foreman. They get the RNA, that's the blueprints. They're capable of reading the blueprints and getting the required materials to the job site and having the materials be put together in the right way to make a protein. Right? So the ribosomes build the proteins using information given to them from the nucleolus. That's the extent to which you need to understand the process of protein synthesis. The nucleolus copies the DNA, sends the RNA to the ribosome builds the protein. You know that much, that's good enough for Science 10. In Bio 30, you'll go a lot more into the biochemistry of protein synthesis, and you'll learn about all these enzymes and amino acids and all of that, okay? But you'll learn all about that later. For us, as long as we understand, okay, that the nucleolus copies the DNA and the ribosomes make the protein, which is, okay, so ribosomes, okay, which actually are produced in the nucleolus, make the proteins. Okay, so we're getting two things off that slide. One thing we already kind of had, okay, the nucleolus makes the ribosomes, okay, and the copied DNA, and the ribosomes make the. Ribosomes make protein, and they make it using single strand of genetic material that comes from the nucleolus. Okay? If you know that much, then you're good for what the ribosome does and what the nucleolus does. Okay, can we move on from that slide? Okay, now in your cell there are two kinds of endoplasmic reticulum. When we labeled your diagram, there's smooth endoplasmic reticulum and there's rough endoplasmic reticulum. You don't always have to use endoplasmic reticulum. You can write ER and I'll know what you mean. Okay? So they actually not only look quite different, but they actually do very different jobs. Okay? The rough ER is almost exclusively located right up against the nucleus. Okay? The reason it looks rough is because it's covered in ribosomes. What was the job of the ribosomes? They make protein. So would it be smart if the things that make protein are really close to the nucleus where the instructions come from? Okay, here's another smart thing about the location of those ribosomes. They're on the rough ER, whose only job is to transport proteins. Okay, so the rough ER, and just because this isn't in here really well, okay, the rough ER transports protein. Now, rough ER is fairly organized and uniform looking. Okay? It's always packed up against the nucleus, and it's kind of always folded over itself a whole bunch of times. Okay? Under an electron microscope, it actually looks like this, almost like just layers of tissue. Okay? So that's what it looks like. Every one of those black dots is a ribosome. Okay? It looks quite orderly, okay, because it's, those ribosomes are on there, they're very busy, they're making proteins, the rough ER is taking it away. The smooth ER, by comparison, is nearly impossible to see. I think I told you that yesterday when we were labeling the diagram, because it's clear, it doesn't have any ribosomes on it, doesn't pick up stain, okay, and so it's hard to see, but it actually runs everywhere and it's like completely random, right? It just kind of goes everywhere, like if you took a bunch of spaghetti and just plopped it on a plate, it would just be all over itself, intertwined, entangled. That's kind of what the smooth ER looks like, where the rough ER would be more like uncooked pasta. It's all just kind of orderly and kind of stacked. Okay. All right. So rough ER transports protein. Your smooth ER has actually lots of jobs. Okay. Some more important than others. Okay. The first and probably most important job of the smooth ER, okay, is to make lipids. What's another name for lipids? Fat. Okay, so lipids are made there, and that is in order to build or repair membranes. Okay, membranes, all the membranes of all of your cells are made of fat. Okay, they're a phospholipid bilayer. That means there's two layers of fats sandwiched together, okay, to create a seal around the cell. Because membranes are like a border, okay, they're always getting damaged. Stuff's hitting them, breaking them, there's chemical things that go on. So they always have to be repaired. So your smooth ER makes the membranes that are used to repair, or makes the lipids, sorry, that are used to repair the membranes. Probably its most important job. Okay. The second most important job of the smooth ER is that it is kind of like the cell's liver. It's going to break down toxins and drugs and things like that that might end up in your cell. Okay? I mean, we consume things that are not good for us all the time. Okay? And our bodies, as they naturally carry out our business, also create toxins that have to be broken down. Right? As an example, if you work out, okay, you're running really hard or lifting weights, lactic acid will begin to build up in your muscles. Lactic acid is a toxin. Okay, that's why it burns and why you can get lactic acidosis that would actually make you pass out. Okay, it has to be broken down within the cell. Okay, if you were to consume alcohol or nicotine or other drugs, okay, those things, they're toxic. Your cell has to break them down into less toxic materials. Right, in the same way that your liver does on a larger scale. Okay? Your liver cells are specialized. They have way more smooth ER in them than any other cell in your body, because their main job in the liver is to break down the awful things that our body produces or that we may ingest. Okay? Now, it doesn't make it all perfect. Okay? It breaks down the toxins for sure. Okay? That's part of it here. Okay? So drugs and poisons are detoxified, okay? But they're made less toxic. They're not made completely not toxic anymore. Okay, so let's say you're an adult and you go out and you, let's say overindulge a little, okay, with your friends. and you've consumed a little too much. How do you feel the next day? Not good, like hungover, okay, is usually the term we use to describe it. That feeling is actually the result of your body breaking down the alcohol or whatever other toxic material you consume. Okay, it's breaking it down. It's making it less harmful to you because alcohol is poisonous. Okay, you can get alcohol poisoning that basically shuts your body down. Okay, if you consume way too much. If you consume, moderate amounts, you can still not feel great the next day because your body makes other things that it can get rid of. Okay, so that may include that you have a headache. Maybe you're nauseous, maybe even to the point of vomiting. Okay? Those are all ways that your body has broken something down and now is helping you get it out. Okay? Your body is designed to get the things that are not good for it out of you. Okay? And that's the smoothie R, making things that are less toxic, but toxic enough that you'll want to get rid of them. Okay? Making sense there? All right. Okay, 2 less important jobs of the smoothie R. So I'd be mostly concerned with you knowing the repairing of membranes, so making lipids, repairing membranes and detoxifying poisons. Okay, but other things that it can do is it can break down complex carbohydrates. Okay, I don't want you to mistake this for digestion. That's different. Okay, digesting food is a different process. Okay, but complex carbohydrates, which we often refer to as starch, okay, are really just simple sugars that are chained together. All right? So it's quite simple for your smoothie R to simply break the chains so that you have a lot of simple sugars. So if you went home tonight and you had like a plate of mashed potatoes, or you had a plate of pasta or a big bowl of rice, okay, you've consumed a whole bunch of starch. Okay? There's a lot of energy in starch. And when it gets broken down, it gets turned into sugar, and your body can readily use that for fuel. Okay? The smoothie R can do that. It doesn't have to wait for the lysosomes to digest starch. The lysosomes can work on harder to digest things like fats and proteins. Okay? Making sense there? Okay, last thing, and it does very little of this, it's why we don't mention it very much, is that certain types of fat-soluble hormones can also be produced here. Pretty minor one. It's here because it does it, not what I'm going to look for. Okay, on a test, I would expect you to be able to write one of the 2 big ones. Okay, one of the 2. If I was asking you, what is the job of the smoothie R, and you actually had to write it out, just one would be fine. Multiple choice question, that might have 2 or 3. Okay. All right. Questions on the ER, smooth and rough? Okay. Okay, so lipids, we just talked about those. Okay, because the smoothie R runs everywhere, it has access to all parts of the cell, so it can rapidly repair any damaged membranes. Okay, Golgi apparatus. Okay, the other day when we went over the labeling, I compared the Golgi apparatus to the Amazon sorting center, okay, that is in Balzac, right? They're right next to Cross Iron Mills there. All right, your Golgi apparatus works just like that building does. Stuff comes into it, okay, that would be the receiving department, okay, stuff comes into it from other places. That stuff could be waste. It could be materials that cell produces and needs to export. Okay? Those things would come in on one side of the Golgi. Okay? Now, you don't need to know the 2 sides of the Golgi. Okay? The cis face and the trans face, you don't need to know those terms. Just know that it's got 2 sides. Okay? So material comes in on one side, and then the Golgi will package it. All it does really is put it in a vesicle, a little bubble. That bubble is made of membrane material, okay, phospholipids. That vesicle, that bubble, then goes towards the cell membrane, and when it merges with the cell membrane, it just becomes part of the cell membrane, and whatever was inside just pops out. Okay, and now it's out into the bloodstream or the interstitial fluid or whatever. It's out of the cell. Okay, that's exactly what the Amazon sorting facility does. Big semi-trucks come into the receiving side of that building, and they bring all of the goods from the manufacturers that sell things on Amazon. Okay? So all sorts of things come in there, from your little trinket junk stuff that you buy to like a computer or something like that. They all come in on trucks. Okay? Once they get taken off the truck, the people inside the building have to process them. They have to go, okay, this package or this product needs to be packaged in a box. This label needs to be put on it so that it can go to so-and-so's house at this address. Then, once it's in the box and it's got the fancy tape and the label on it, it gets put in one of Amazon's gray vans. Okay, and then those gray vans go to your house and drop it off on your doorstep. Okay, exactly what the Golgi does. Okay, stuff comes in, whether it's stuff for export or waste, it gets packaged, it goes out the other side and out to the membrane. Okay, exactly the same kind of thing. It's in shipping and receiving. Okay. Okay, so shipping and receiving gets rid of waste. Okay, something like that would be, good. Contents of the vesicles are modified for transport out of the cell. So it takes things and it gets them out. Right? That's its job, is to get things out of the cell. If you look at the Golgi under the microscope, it is quite obvious that it has two sides. They look very different. Okay? In this diagram here, you can see that the cis face, that's the receiving side, is quite smooth. But the trans face, where the stuff is being shipped out, okay, that's quite rough. And that's because it's constantly budding these little vesicles that have the stuff that it's processed in it. Okay? Under an electron microscope, it looks like this. So these dark little folded lines right here that kind of look like a smile, okay, that's the Golgi apparatus. On this side, you see not a lot of bubbles, okay? You just see kind of material coming in. But here, on the shipping face, okay, we can see there's tons of bubbles on this side. This is the side that's shipping the vesicles out to the membrane. So when you actually see it under the microscope, and you can sometimes see them through our microscopes in our lab, you'll be able to tell one side from the other, because it does kind of curve, okay, like a smile. Okay, any questions on the Golgi? Yeah. All right, lysosomes. Job of the lysosome, digest food. Therefore, they're only going to be in what kind of cell? Plant or animal? Animal. Okay, lysosomes are an animal-only organelle. Everything else we've talked about, it's in both. If you have the diagram like this, if you have the printed notes, okay, I did make a couple of changes to it. Okay, on your diagram, this said lysosome right here, and I scratched it out and put in peroxisome, and then I put lysosome up here. It's a small distinction, but if you want to have the correct diagram, okay, you can have it. It's not something I'm really going to test actively. So a lysosome is where digestion occurs. It has digestive enzymes that are capable of breaking down cellular material and converting it or putting it into different forms, usually sugar. Okay? In order to do that, okay, there has to be digestive enzymes in there. So the digestive enzymes are actually one of the things that comes into the Golgi and then is repackaged and comes out as a vesicle. When that vesicle full of enzymes comes out, it's not a lysosome yet. It's a peroxisome. We talked about the difference there when we did the labeling. Okay, so it's called a peroxisome. It's just a vesicle containing the enzymes. It becomes a lysosome when it merges with a food vacuole. Okay, now the enzymes are actively digesting something. Now it's a lysosome. In the end, it's pretty hard to tell under a microscope which one is which. Okay? In fact, most of the time it's pretty hard to see lysosomes, period. Okay, so as long as you know the lysosome is responsible for digestion. It can also be responsible for recycling. Okay? Every time a cell divides, some of the organelles that were in the original cell are carried over into what we call the daughter cells. Some of those organelles could be 20 generations old. Okay? They could be from a cell that's divided, the original cell 20 divisions ago. Organelles don't last forever. Okay? They break down and they stop working. At that point, your cell is the ultimate recycler. It just sends a peroxisome over, merges with that organelle, and digests it. Okay, then the raw materials can be used to build a new one. Okay, making sense there? Okay, there's one other job of a lysosome. Doesn't have to do it very often, but it's kind of an important job. The lysosome is like a fail-safe. It can act as a self-destruct mechanism for a cell. The biggest problem a cell can face is if its DNA becomes damaged and then becomes malignant or cancerous. Okay? When that happens, the cell begins to divide rapidly, out of control, and that's what forms the tumor. Okay, so if you have a tumor, that's your own cells, but they've just been dividing rapidly and they just get bigger and bigger. The problem with cancer is those cells still require lots of nutrients and they eventually kill the healthy cells around them and they're malfunctioning, so it's not good. If it's growing on a major organ, okay, that's not good for you and the tumor needs to be removed or killed. Okay, so there's two different ways to kill a tumor. Radiation, so you just focus radiation on it and kill the cells, okay? Or chemotherapy, which will kill all rapidly dividing cells in your body. That would be why your hair would fall out and things like that. Okay, so when a cell has the ability to detect if it has become malignant, it's a limited ability, but it can. If it detects that, it releases a chemical that basically dissolves the membranes of all the organelles inside, including the lysosome. If the contents of the lysosome get out into the cell, what will they do? Digest it. Okay? The cell essentially eats itself. Right? That would kill the cell and it would prevent it from becoming malignant, dividing out of control and becoming cancerous. That process works most of the time. Okay? It's why cancer is not super common. Okay? It can happen. It usually happens when the part of the DNA that codes for that mechanism is the part that's damaged. Okay, so for the most part, lysosomes are digesting food. And they do have to have a pretty robust membrane because we don't want that stuff getting out into the rest of the cell. Okay. All right, questions on the lysosome? Yeah. All right, mitochondria. Okay, in junior high, you were probably told this is the powerhouse of the cell. And it is. It does generate the energy the cell needs by carrying out this process. And so if you're going to write like one thing about the mitochondria, it would be that it carries out cellular respiration. Okay, so cellular respiration is the process that takes glucose, reacts it with oxygen, makes carbon dioxide and water, and releases the energy stored in the glucose. All right, that process or that reaction looks like this. What kind of reaction is that? It's a combustion reaction, exactly. Okay, cellular respiration is a combustion reaction. Don't get the idea there are tiny little microscopic fires going on inside your cell. Okay? Yes, it's like a combustion reaction, but it's not burning. Okay? It is releasing the energy, and its byproducts are going to be carbon dioxide and water. Okay? Now, the mitochondria has a double membrane. We haven't talked about any other organelles that had a double membrane. Why would it be important for your mitochondria to have two membranes? Yeah, it's kind of insulative. Okay? If cellular respiration is going on in there, that generates heat. Okay? It's why now and for the rest of the semester, I will be using you to heat the classroom. Because they came in here in period 4 yesterday and closed the valves for the heating. They were stuck open. Okay? That's why it was so hot in here. Okay? The hot water heating was just running through and it wasn't shutting off. Okay? So now I'm going to use the little tiny microscopic mitochondria that exist in numbers in the trillions in every one of your bodies, okay, to keep the room warm. I've been doing it for like 20 years. It works. Okay, it's cool in here in the morning, but by afternoon, it's okay. Right? They generate a lot of heat. that reaction, that cellular respiration makes a lot of heat. It's why your body runs at 37 degrees Celsius, or why when you have a fever, it runs hotter than that. Okay? It just means that when you have a fever, those mitochondria are running hotter. Okay? They're burning more sugar, okay, in order to drive up your body temperature to hopefully weaken whatever has made you sick. If you work out, same thing. Okay? You're demanding more energy when you're exercising or running or whatever you're doing. Okay? So it's generating more heat. You sweat. Okay? All of that kind of stuff. Another weird thing about the mitochondria, you can see it on this diagram here. Mitochondrial DNA is labeled. Mitochondria have their own DNA. Mitochondrial DNA exclusively comes from your mother. Okay? Why? Anybody know? Because when the sperm and the egg meet, the egg represents the bigger part of that single cell that divides over and over again to create you. Okay? There's very little in terms of mitochondria in a sperm. They don't survive very long. So most all of your mitochondrial DNA is also maternal. Now, mitochondria have their own DNA because the theory goes that mitochondria are actually organisms that were absorbed by bigger cells and then began to work together collectively. Okay? The idea of the first eukaryotic cell is actually not so much that it evolved, but more that one type of cell absorbed some small prokaryotic cells, and they just continued to work together because it was to mutual advantage. So the DNA that's in your mitochondria doesn't code for you. It codes to make a mitochondria. Okay? But it's different. It's actually another thing that criminologists will use in identifying people because it's even more unique, okay, than your nuclear DNA is. Okay. All right. So yeah, double membrane. The double membrane's insulating. Okay. Cellular respiration goes on in there. Questions on the mitochondria? Okay. Chloroplasts only going to be in plant cells. Okay. And what I've circled here, or highlighted here in yellow, a chloroplast carries out photosynthesis. That is its one and only job. Now, the chloroplast also, the chloroplast also has a double membrane. Okay? It's not that photosynthesis itself produces a lot of heat, but if something absorbs a lot of light energy, is it naturally going to get hotter? Yeah. If you absorb energy of any kind, your energy increases. That usually becomes thermal. You can kind of think of it this way. Like if you've got a black car and a white car in a parking lot on a hot sunny day side by side, which one's going to be hotter to the touch? The black car. Black absorbs more light than white does. White reflects more. Okay? So anything whose job it is to absorb light energy is naturally going to get warmer, okay, as that process goes. So it also has a double membrane. Okay? And inside the double membrane, you might even be able to see these through the microscope next week, will be these little stacks. Okay? They look like stacks of like checkers or coins or poker chips or something like that. Okay? And that is where photosynthesis takes place. It's where that whole thing about the electrons getting excited and jumping up levels, and then when they fall back down, they release the energy. That's where that occurs. Okay? Is in the thylakoid. You might even be able to see those if you get a chloroplast really well focused. Okay? And chloroplasts will be readily visible in the plant cells. Okay? Questions on the chloroplast? It's a pretty easy one. Carries out photosynthesis. Good, done. Check that box. All right, so we got a few other ones here that we need to talk about. So what I'm going to do is kind of give you a chance to write down the highlighted stuff here, and then I'll talk more because then you'll hear more of what I say. Okay, so anything that's underlined in yellow, okay, that would be the important stuff. I know, like I've got a box around the word frame here. maybe do that too. You'll know why that's important when we talk about it. Okay, I'll give you a minute to write that down. Okay, so we'll first talk about the cell membrane. Every diagram you ever get of the cell membrane makes it look like it's this kind of featureless layer that's just nice and smooth. Okay? Nothing could be further from the truth about the cell membrane. Okay? It is nothing near featureless or bland or smooth. Okay? In fact, it's very busy and rough. Okay? Most people get the idea like the cell membrane is like jello, right? Just smooth, you can see through it, there's nothing to it. It's actually more like jello salad. Okay? Jello salad is when you put all kinds of fruit inside the jello when you make it. Okay? So you got little pieces of pineapple sticking out, or a grape in there, or whatever else. There's all kinds of things in the jello salad. That's what the cell membrane actually looks like. Okay? So this diagram here, okay, is actually what the cell membrane looks more like. Okay? So you can see here that there's all kinds of stuff in the membrane. Yes, there's that phospholipid bilayer, the fat layer that is most of the cell membrane. That part would be fairly smooth. But there's all kinds of other things sticking out. Yeah. Okay? There are globular proteins. There are receptor molecules, glycoproteins. Cholesterol is embedded in that layer. Okay? It's an important part of the cell membrane. Okay? Glycolipids, okay? Oligosaccharide chains, all kinds of stuff. It's a very complex, very busy place. All right? It's got a lot of things going on. And it also has special proteins that stick out. Some of those things are just to receive chemical messages. Okay? They would indicate that maybe a certain type of hormone has landed on the membrane of the cell, like maybe adrenaline. That's a chemical messenger you kind of need to take into the cell so that it can do its job. Right? So there's lots of little antennas kind of things all over the cell membrane. Weird thing about the cell membrane, it needs a different antenna for every signal it can receive. Okay? It's not like the antenna in your car. Okay? One antenna receives every station. Okay? Your car's antenna is one antenna. This has to have an antenna for every station. So it's busy, right? There's all kinds of things sticking out of this layer. And its main job, if you're going to write what the function of the cell membrane is, these are the two words I want you to put on there. Endocytosis, movement in. Exocytosis, movement out. Okay, those are the two words I would want you to write if you were giving me the function of the cell membrane on a quiz or a test. Okay. Now, the cell wall, same idea. Okay? The cell wall also, when we use the term wall, we picture something like, the four sides of this room. It's smooth. I can't walk through it. Okay? It's a barrier between the inside and the outside. Nothing could be further from the truth. Okay? In a plant cell, at least, the cell wall is more like a frame. Okay? You've all seen a house being built, right? Right? When they first put up the walls, you can walk through the walls. The house is only framed. Okay? Though that frame can hold up the house, it's very strong, but it's not a barrier between inside and outside. The cell wall doesn't protect the cell at all. It's not a wall. It's a frame. Okay? Everyone kind of follow the difference there? And you can see here, okay, this green stuff, that's the cell wall under an electron microscope. You can see that it's hollow. And you can get through this way and this way. It actually looks kind of like the wooden framed walls of a house. It's just there for support. It helps to keep the cell's shape, but it doesn't protect it in any way, shape, or form. Okay? And as we talked about the other day, okay, it is made of cellulose, which is plant fiber, okay, or what we call dietary fiber. We talked about that the other day about how we can't digest that, and it just goes right through us, but some herbivores can, because obviously they eat plants for food, they have to be able to break it down. Okay? Our digestive system can't do that. All right, so for cell wall, I would say you need to have that it's a frame and that it helps keep the shape or support the shape of the cell. It cannot do that job on its own. Okay? It has to work together with the next thing on the list, and that's the water vacuole. Okay? So inside of the cell is this big kind of bladder that's full of water. Okay? When that bladder is full, it pushes everything in the cell against the frame, making it stronger. Kind of imagine this. You and a bunch of friends are in this room, and in the middle of the room is this big water bladder, and someone is filling it up. Okay? It's going to get bigger and bigger and bigger, and eventually you're all going to be squished against the walls. It'll exert pressure against the walls, and you'll right up against the wall like that. Okay? That's how the water vacuole works. It pushes from the inside against the frame, making it stronger. That's why if you haven't watered a plant for a while, it wilts. We talked about that the other day as well. Okay? When that water vacuole can't push, the cell wall will fold. Okay? It isn't strong enough by itself. Okay, so the way it does that is it uses turgor pressure, fancy word for the water pressure. Fancy word that you need to know. Okay? Turgor pressure is water pressure from within. Okay, so obviously cell wall and water vacuole only present in the plant cells. Okay? Animal cells don't have cell walls. Okay? Fungi have cell walls. Okay? Bacteria have cell walls, but they're different. Okay? A fungus cell wall is actually made out of chitin. It's the same thing that the exoskeleton of a bug is made out of. Okay? So if you've ever stepped on a bug and you hear that crunching, okay, that's chitin. Okay? It's very hard, it's very stiff. Okay? That's what the fungal cell walls are made out of. Bacterial cell walls are typically made out of things like peptidoglycan, things like that. That's the chemical that penicillin reacts with. Okay? So when you take penicillin because you have a bacterial infection, it actually breaks down the cell wall of the bacteria, okay? And that will kill it. Okay? All right, next thing, centrioles. Now, centrioles are only in what kind of cell? Animal cells. We talked about that yesterday. Okay? Their job is to make sure that cell division happens reliably. Plant cells don't need them because plant cells don't change their shape. They got a water vacuole and a cell wall that keeps them rigid and rectangular, okay? And they don't have to worry about anything interfering with the DNA moving to opposite ends of the cell. An animal cell needs to make sure that all the DNA goes where it's supposed to because it could twist or bend or stretch during the cell division process, and some of the DNA might get lost. So, when the cell is about to divide, the first thing it does is copy all of its DNA. Then, the DNA coalesces. Okay? Inside your nucleus, the DNA doesn't look like chromosomes like this. Okay? There are chromosomes in there, but they're not organized like this. Okay? Chromosomes in a normal cell, and DNA in a normal cell is akin to this. You buy a ball of yarn, and you completely unravel it and throw it on the ground in a pile. That's what your DNA looks like most of the time while your cell is actively doing its thing. When it's about to divide, it gathers it all up and organizes it again, because it's easier to keep track of everything then. So it'll gather it up and make the chromosomes line up right along the middle of the cell. And then the centrioles will split, and one will migrate to each end of the cell and copy itself. Once it's copied itself, it sends out these little fibers that will grab onto the chromosomes and pull them apart before the cell completes its division, making sure that all of the DNA gets where it's supposed to go. What do you need to know at a bare minimum? Centrioles help animal cells with division. Okay? That's what you need to know. Okay, and last things on here. Okay, microfibers and microfilaments. Sorry, you couldn't see that before. I had to scroll down for you to see it. Okay, microfibers and microfilaments are mostly an animal cell thing, and they help maintain the structure of the cell so that it doesn't tear when it's changing shape. Okay. We sometimes call it a cytoskeleton, which is kind of a bad name because it implies bones, and they're not bones. They're flexible. They're like bungee cords. Okay, so inside an animal cell, and this is not something you'd ever see with our microscopes, there are these microfibers, microfilaments, and they just latch on to different parts of the cell membrane. When the cell changes shape, they can stretch, okay? And they'll usually keep the cell from tearing. Not always. Okay? What cells in your body do you tear all the time? Muscle cells. Okay? It's how you get stronger. Okay? You go and work out, what you're doing is intentionally tearing your muscle cells. Okay? And then they'll grow back, and they'll grow back stronger. Okay? One of the ways that muscle cells are specialized is they are multinucleate, which means they have more than one nucleus. That way when they tear, it's likely that both sides on the tear would have a nucleus and they could grow back, okay, and be stronger for it. Okay? So they have these fibers. When you break them, they'll go back together. Okay? They'll grow back and you'll get stronger. Now, depending on what type of exercise you're doing, you will build and repair those things differently. If you're lifting really heavy weights because you're trying to bulk up, okay, those things will grow back together, but they'll grow back a lot bigger. Okay? You'll gain muscle mass. If you're just trying to get stronger, you usually lift like light to moderate amounts of weight, and when they grow back, they simply grow back stronger, but not a lot bigger. If you're trying to increase your endurance, you're breaking different types of muscle fibers, okay, they'll grow back, but they won't get bigger at all, okay? They'll just grow back and be able to last longer, okay, as opposed to tear again, okay? Muscle that's about super strength, okay, tears easily. Right, so it's why you can't do lots of reps with that kind of exercise. If you're trying to put on muscle mass, you do very low numbers of reps with a huge amount of weight. If you're trying to build endurance in your muscle, lots of reps with very small amounts of weight. You'd be, with those little tiny dumbbells, this big. That's what you would do, right? You don't really break it very much. You just make it capable of repetitive movement. Okay, as opposed to when you're just trying to get huge, okay, you're tearing lots of stuff and you're just like, I got one, sweet, I can put it back on the rack. Okay, works kind of like that. Okay, questions on any of those? All right, I'm going to give you about a 4-minute break here before we start talking about the transport stuff. All right, so now we got to talk about cellular transport. And we start out by covering the big three. Okay, the big three methods of cellular transport. Okay, diffusion, which is how any cell transports material from one place to another within the cell. It's intracellular transport. Okay, we'll talk about osmosis, and we'll talk about active transport. Those are the big three methods of cellular transport, the way things move in and out of the cell, and the way they move once they're in the cell. Okay? Diffusion is the most common form of cellular transport, okay? And it's how everything gets from point A to point B within the cell, okay? And things have to move from point A to point B within the cell, right? There's no bloodstream or vessels or a heart within the cell that's moving things around. They just have to move on their own by diffusion. Now, diffusion is a natural process, and it is also passive. So you'll see that underlined here. It is a form of passive transport. That means it doesn't require any energy from the cell to make it happen. Now that makes it sound like it's really good. Okay? It doesn't cost the cell any energy. And while that's true, it's not without its drawbacks. You kind of get what you pay for with free. Okay? So diffusion will reliably move material from an area of high concentration to an area of low concentration, or what we call down the gradient. So you'll see here, okay, that I kind of drew this diagram here. Okay, high concentration, low concentration. The difference between is the gradient. It's kind of like a spectrum, right? There's a spectrum of there's people at this end and people at that end, and there's kind of a varying change of grades between one end and the other. Gradient and the spectrum are kind of the same thing. Okay, so the high concentration is the top of the gradient, low concentration is the bottom of the gradient. Everything naturally moves from high to low. Okay? The reason for that has to do with the particle theory. Okay? All particles are always in motion. Okay? The higher their temperature, the faster they move. So when things are crowded in a small space, that would be high concentration. They want to move, but they don't have a lot of room. So they naturally spread out because of the natural motion of their particles. So in this picture here, we can see that dye is being injected into like a pool, right? Here at the injection point, the concentration is very, very high, but it's naturally dispersing out from that injection point. Okay? Diffusion it works on the same principle as a fart. Okay? If somebody farts and you're right next to them, your experience is different than the person on the other side of the room. Okay? Because you are at the area where the fart molecules are in highest concentration. Okay? And so your experience is quite intense. The fart molecules will naturally spread out throughout the room. They will diffuse because all particles naturally do this. They will move from an area of high concentration to low concentration. If you're on the other side of the room, you may never even know that a fart has occurred. Because by the time that molecule gets over to you, it's spread out so much that it's virtually undetectable. Lucky you. Okay? Not so lucky for the person right beside. Everyone kind of follow? Yeah, I use a fart because nobody forgets. If I talk like perfume or something like that, people forget. Fart, no one forgets. Okay? So that's how diffusion works. It naturally moves from high concentration to low concentration. But as it does that, it spreads out. So if I'm using diffusion to move something from an area where it's being produced to an area where it's needed, how much of it's going to get there? All of it, or some of it, or none of it? Some of it. Okay? This is not like targeted delivery. Right? This is a shotgun approach to cellular transport. Okay? Does everyone know what I mean when I say shotgun approach? Right? When you shoot a shotgun, the pellets come out and they spread out and they get further apart. That's why you use it for duck hunting. Okay? You don't try to shoot down a goose while it's flying over your head with a single bullet. You got to be a really good marksman. Okay? That's why they use shotguns. All this shot, the little pellets fly out, and you got a much better chance of hitting the goose. Okay? So a shotgun just means that the stuff spreads out over a wide area. So the thing I'm producing might be right here, but the place it needs to go is here. So here, it would be very, very concentrated. But it's going to kind of spread out through like a three-dimensional cone on its way towards its destination. Or not so much its destination as it moves down the gradient. Well, by the time we get to this area here, I have the same number of dots. But maybe 2 or 3 of them is actually getting where they're needed. Okay? And that gets worse the farther they have to go. Diffusion is really good as long as it doesn't have to go very far. Okay, when we talk about the fart in the classroom, that's a long distance for diffusion to work over. That's why we notice a huge change in concentration from one side of the room to the other. But if we're talking about a cell is really, really small. So diffusion is okay because it only has to go, what, at the most, maybe 100 micrometers? Okay? The diffusion cone is not going to be a huge problem as long as the cell's not overly big. The activity we're going to work on tomorrow is going to look at what is the effective size on the ability of a cell to use diffusion. There comes a point where a cell gets too big and it'll literally starve itself because diffusion just doesn't get enough stuff where it needs to go because it's had to travel too far. Okay? Now, diffusion is also not really fast. It's like free shipping, right? If you pay for the shipping, you got it the same day or the next day. If you get the free shipping, 2 to 5 days. Okay? It's not super fast. Okay? All right, so simple rule of diffusion, okay, is that in the absence of anything else, like a current or anything, okay, substances will diffuse from high concentration to low concentration down the gradient. It is passive. It doesn't require any energy. Okay? So it's just going to happen. That's the other drawback to passive transport. If it's passive, it's going to happen, whether you want it to or not. Okay? You cannot control it. It is just going to go. Okay? But diffusion works pretty reliably, okay, for most cells. Okay, any questions on diffusion? Okay, diffusion is the most important one. Now, with the cell transport stuff, these are going to be your big application questions. So yes, you need to know what diffusion is and how it works. That part you have to memorize. But know that I'll test it by giving you a situation. Here's a cell. It's in this situation. Identify which of the cell transport processes covered in class would be most applicable to here. Explain how it works and how it applies to this situation. Okay? That's how you'd be tested on this stuff. I mean, yeah, you might get a multiple choice question. It's like just the definition of diffusion, but I will ask you application questions like that. Okay? All right. The second passive of the big three cell transports is osmosis. Now, osmosis is quite a bit different than diffusion. Diffusion can transport anything. Okay? It can transport ions, minerals, vitamins, all kinds of stuff. Osmosis is exclusively water. If something is undergoing osmosis, only water is moving. Okay? And it's not within the cell. It's across the membrane. Okay? So as I said, these are two very different things. Diffusion happens within the cell. Osmosis happens from the outside to the inside, or vice versa. Okay, I'll let you underline the parts that are important. So osmosis, passive transport of water across the membrane to balance salt concentrations. Okay, that would be like your basic definition. Anybody ever have a leech latch onto them? How'd you get it off? Salt. Yeah, you can pull it off, but you'll bleed a lot more. Okay, the better way to get a leech off of you is to pour salt on it. And the same thing will happen to it as happens to a slug. Okay, hopefully no one's done this, where you just pour salt on pour in some little slug. Okay, but if you pour salt on a slug, what does it do? It shrivels up. And it shrivels up because its skin is different from ours. Okay, ours is a pretty good seal against the outer environment. Okay, but if you put salt on a slug, their outer membrane is fairly moist, and so that salt will immediately begin to draw water out of the slug by osmosis. Okay? Osmosis wants to balance salt concentration. So if I throw a whole bunch of salt on that leech or that slug, okay, all the water that's inside the slug starts rushing towards the salt so that the concentration outside and the concentration inside are the same. Now for that poor slug, not the poor leech because there's something in your blood, okay, but for that poor slug, okay, that is never going to happen. There's not enough water inside of that slug to counteract all of the salt that's poured on it. So it just rushes out and the slug just shrivels up. Okay, osmosis is always going to have water move towards salt because it wants the concentration on both sides of the membrane to be equal. The same thing can happen for you. If you're stranded in the middle of the ocean, should you drink the seawater? No. And there's two reasons for that. If you remember Finding Nemo, okay, all pipes lead to the ocean, which means the ocean is the world's toilet. Don't drink toilet water. Rule number one. Okay. Second reason, it's salty. Okay. It's saline. We don't want to consume that because if I drink a whole bunch of salt water, now my blood gets salty because that salt has to go into my blood. And if my blood is salty, water from the rest of my tissue is going to naturally, passively, and without my ability to control it, move into my bloodstream, okay, in order to equalize the salt concentrations. When that happens, my kidneys will immediately detect that I have too much water in my blood, and they'll make me urinate out the water. If I drink a liter of salt water, I'll probably lose 1.5 liters of my own bodily fluids to help osmotically balance it. Okay? It's why you can get dead in pretty short order by drinking salt water, by drinking ocean water. Okay? It's simply osmosis. It's not something that you can control. Okay? In fact, for the first couple of days that you're stranded on the ocean, you're actually better off to drink your own urine than the salt water. It has less salt in it. Okay? That osmotic balance won't be so bad. I mean, it'd be bad to drink urine, but it won't be as bad as you'll live longer, okay, than you would if you drank salt water. Okay? I know I just said the ocean is the world's toilet, drink your urine, but okay, you get the idea. Okay? It's the difference in salt concentrations there. Okay? Restaurants and bars will use this as a trick. Lots of times, if you're at like the bar part of a restaurant, there'll often be like free pretzels or popcorn. Both of those foods are salty. Okay? So you eat the salty food. Okay? The salt goes into your bloodstream and pulls water out of your tissues and makes you feel thirsty, and you buy more drinks. Okay? Pretzels are cheap. You can buy them in huge boxes. Okay? But you're buying more and more pop or whatever it is you're drinking. Okay? You're drinking more and more of that because you're feeling thirsty because you're eating the salty stuff. Okay? Appetizers at a restaurant, almost always salty. Why? Because you're going to buy more drinks while you eat. Okay? It's insidious, but it's also clever. Okay? And it simply uses science. Okay? Here's another one for you. You guys remember sometimes we get that gospel reading about putting the new wine in old wineskins? You don't want to do that because the wineskins will burst. That's osmosis. If you put wine into a wineskins were actually made out of like sheepskin. Okay? So you'd make these things that were like a flask. They were sealed up like sheepskin. As the wine stays in there, a lot of the salts from the wine will be absorbed by the skin. Okay? That's not a big deal as long as you don't change the wine. If you then put fresh wine in there, there's more salt in the skin than there is in the wine, and the water will move from the wine into the skins, and the skins will expand and burst. Okay? Science. It's even in the Bible. Okay? It's just osmosis, but that's why it happens. All right, so this is actually an experiment here that you'll probably do in Bio 30 when you're doing the kidney units. Okay, you're doing excretion and talk about salt balance within the kidney. Okay, you'll do an experiment where you have kind of a horseshoe-shaped test tube. In the middle of it is a place where you can put a piece of sausage casing. And a sausage casing is just the intestinal lining from a pig usually. Okay, so you put that across there, you now have a cell membrane. On one side, you'll have tap water. And on the other side, you'll have tap water that you've added salt to. Over the course of even a few minutes, you will start to see the water level on the tap water side drop and the water level on the salt side go up. And it'll continue to do that until the concentration on both sides of the sausage casing are equal. It's osmosis. The water is moving to the salty side in order to balance the concentrations. Okay? So it's not something that your cells can control. It's passive, so it's just going to happen. That's why the slug dies and the leech dies, okay? Is because they simply can't stop the process. All right, so if I put a cell in water that's basically the same concentration as its cytoplasm, equal amounts of water will move in and out by osmosis across the membrane, and they'll be nice and balanced. Okay? If I put a single cell into salty water, the cell will shrivel up. Just like the leech, just like the slug. Okay? The water will move out of the cell and into the salty water. That doesn't mean there aren't salts in the cell. There are, but there's more outside. If I put a cell in distilled water, the cell will pop. Because there's salts inside the cell. There's no salt in distilled water. So the water just rushes across the membrane by osmosis into the cell because there's salt in there. And the cell will just keep getting bigger and bigger and bigger until it pops. Because there's no way to balance it when there's none on one side and some on the other. Okay, making some sense? Okay, so osmosis, passive movement of water only across the membrane to balance salt concentrations. That's what you need to know. Okay? Okay, osmosis and diffusion are both passive transport. The last of the big three is active. That's why we call it active transport. It actually uses energy to make it work. And it's because it's forcing stuff up the gradient, not allowing it to passively fall down. Okay, so while diffusion is more like letting a river run its course and the water flows downhill, active transport is pumping water back up to the top. that would require energy. Water is not going to naturally go uphill. Okay, so in here, obviously the underlying parts talk about that this is a process that is active, that requires energy. It's moving material up the gradient, which is not the way it would naturally go. Okay, that is accomplished by what you see down here at the bottom. Okay, I've circled the words carrier protein. Carrier proteins are some of those things that are the fruit salad, the stuff that is embedded in the cell membrane. Okay, there are lots of carrier proteins, and they can change their shape when energy is applied to them. All they do is kind of pivot, and they open and close, right? So when energy is applied to them, in their resting position, they're open to the outside. Okay? So minerals and things like that will pass, just kind of fall into them. When energy is applied, they'll snap the other way. They'll be closed to the outside, open to the inside, and they'll just keep doing that as long as the active transport is going on. So as they change their shape, they actually create a pumping action that will pull material through the membrane and into the cell. This only moves small things. Okay? We're talking ions, sodium, potassium, okay? Things like that. Active transport is a really huge part of what makes your nerves able to transmit nerve impulses. It's also a big part of what allows your muscles to contract and relax. Okay? There's an active transport called the sodium-potassium pump, okay, that works on both your nerves and your muscles. Okay? So when you contract a muscle, okay, the sodium and potassium switch places. That's what creates the electric potential, okay, that causes the muscle to contract. In order for the muscle to properly relax, those have to be pumped back the other way. Okay? So it takes a little bit of time. That's why, if you're really dehydrated, you can get muscle cramps. You cannot reestablish the sodium and potassium on the right sides. The pump is exhausted, okay? And it simply cannot balance it. The muscle won't relax. Okay? That's what a muscle cramp or a spasm is. The muscle is just tense and you cannot relax it. Okay? If you can reestablish an osmotic balance, then it'll reestablish the ability of the cramp to let go. All right? Now, the whole idea of osmosis is actually being used to develop sports drinks like Gatorade, Powerade, things like that. Right? They put a little bit of salt in those drinks to try and kind of speed up the process of osmosis, but also help establish more sodium and potassium on the right side of cell membranes so that active transport can work as well. Okay? If you've ever been really dehydrated and gone to the hospital and got an IV, what's in the IV? Salt. Okay, very low concentration, like 0.05%, but it's enough that it starts the process of osmosis and getting that water to go back into your cells. Okay, that's why they do it. They don't just hook you up to distilled water. Okay, that's not going to do as much. Putting a little tiny bit of salt in there helps that process to go better. All right, so carrier proteins change shape as energy is applied. That's what helps pump the material across. If you're telling me about active transport, I'd better see a little bit in there about carrier proteins and that they change their shape when the energy is applied. Okay. Now, those are the big three. Those are the ones I'm going to ask application questions on. What we're going to look at here now is transport across the membrane that involves the membrane actually having to change its shape. Okay, so when I told you what's the function of the cell membrane, I said I wanted you to use two words: endocytosis that's movement in, and exocytosis, that's movement out. Okay? Endocytosis and exocytosis involve the membrane having to change shape. Okay? There's 3 kinds of endocytosis. They're mechanically the same, and there's only one type of exocytosis. So exocytosis is those little vesicles, okay, especially the ones that come off of the Golgi apparatus, they just go to the membrane, and they get to the membrane, they merge with it, and whatever was inside just pops out, and that vesicle just becomes part of the membrane. Okay, that's exocytosis. That's either going to get material into the bloodstream, okay, or into the interstitial fluid. That's the fluid between cells. Either way, it's out of the cell. That's all the cell is worried about. I got to get the stuff out. Okay, it's the neighbor that throws garbage over the fence. Okay, so it's just worried about getting it out of the cell and nothing else. Right? So that's how exocytosis works. Okay, it's just like a bag that's got something in it, okay, and you're like holding it shut, and then when it merges with the membrane, you just pop it open and you force out whatever was in the bag. Okay, so the underlying part there would be important. Okay, exocytosis, this is large molecules, by the way. Okay, diffusion, active transport, okay, those were small molecules that could fit through the membrane. This is big stuff. Can't go through the membrane. Membrane's got to engulf it. Okay, so endocytosis is the opposite of this. The 3 different types of endocytosis are only different because of what they're taking in. Mechanically, it's still the same process. Something touches the membrane, and the membrane folds around it and basically turns inside out, and then that material is in a vesicle, or a vacuole we call it, and it's moving into the cell. Okay? That process mechanically is the same for all 3 types of endocytosis. The only thing that makes them different is what's being taken in. Food, water, or chemical messengers, like hormones and things like that. Okay, so there's 3 different kinds. All right, so endocytosis involves, okay, yeah, like if you've ever walked your dog and you have to clean up after them on the walk, right? You put the little bag over your hand, right? You pick up the unmentionable stuff that comes out of them, okay? And then you turn the bag inside out. That's endocytosis. Something touches the cell membrane, it folds in around it, turns it inside out, and it's inside the bag, and you didn't have to touch it, okay? So just like that, that's how endocytosis works, okay? The 3 types: food, water, hormones, okay? So there's phagocytosis, okay? That's when it's a food molecule, okay? Pinocytosis, that's when it's water. And then RME, receptor-mediated endocytosis, that's a hormone. I'm never going to ask you an application question about this. You just need to know which one's happening, like a multiple choice or maybe a numerical response kind of question. Okay, just be able to know, oh, it was taking in food, that's phagocytosis. Okay, oh, it was taking in water, that's pinocytosis. That's all. This is memorize and regurgitate. This is not apply. So we talked about osmosis. We said water could move into the cell by osmosis. Why does it have to gulp it if it can move it in by osmosis? Water only moves in by osmosis if the salt concentration inside the cell is lower or higher than outside. Higher, right? It's always going to move toward the higher concentration of salt. So sometimes if a cell has become dehydrated, okay, it will just gulp the fluid. Okay? It will literally just continually make little gulps, okay, or like chug it, okay, in. It'll just keep making these folds that'll trap fluid, and that'll just come pouring into the cell. Okay? It's kind of a quick way to rebalance that stuff. Yes, it's got to use energy to do it, okay? But it's worth it if the cell really needs the water quickly. Right? This one over here, this is receptor-mediated endocytosis. So we talked about that there's those little antennas all over the cell membrane. If one of those gets triggered, the cell immediately envelops that material and brings it into the cell. Okay? If it's food, okay, there's receptors for food. Okay? A cell knows that that's food, it'll engulf it as well. Okay? So mechanically, all the same. Membrane engulfs something. Okay, so phagocytosis, that's food. It's actually how your immune system destroys bacteria. Okay, the killer cells from your immune system will go along and they'll just engulf bacteria and eat them. Okay, that's a great way for them to get rid of them. Okay, and then pinocytosis, gulping of fluid surrounding the cell. Okay, this is what that looks like under an electron microscope. So you can actually see all those little kind of cups, okay, that have just been pulled in, and it's just going to kind of pinch off there, and there'll be a little vacuole that'll come in that'll be full of water. Okay. And then for receptor mediated, again, you've got the little receptors, the little antennas. They receive their signal, that it triggers the membrane to engulf. Okay. What it looks like under an electron microscope. So here you've got your little receptors. You can actually see I've kind of drawn them on here. Okay. So there's some of them here as well. Okay, there'd be one right here. Okay, kind of looks like that. Okay, and right here, there's one right there. Okay, it's got the molecule. You can see those circular things. Those are the things that are being brought in. Okay, it just triggered that, and it just folds in, and then eventually this just meets in the middle, pinches off. Okay, and in it goes. All right. So mechanically, they're all the same. Endocytosis is engulf. Take it in. All right. Questions on those three? Again, just be able to recognize those. That's all I'm asking for those. I'm not going to ask any application questions about those. All right, perfect. I want you to write down those questions, okay, and do your best to answer them before the bell goes. We're going to talk about those, okay, tomorrow. We're also going to do an in-class activity.